Hi everyone, this is Miss Longley, and today we are going to be looking at osmosis in a more in-depth exploration. We're going to be covering objectives 10 through 11. Number 10 you've seen before with the other two passive transports, simple diffusion and facilitated diffusion. And we'll also cover objective number 11, which is differentiate the terms isotonic, hypotonic, and hypertonic, and explain how solutions of different concentrations can affect cells due to osmosis. So let's go ahead and get started. So to quickly review the cellular transports that we've covered so far, one of them being passive transports, which is moving molecules from an area of high concentration to an area of low concentration. This does not require energy of the cell, so it's naturally occurring and it's by the process of diffusion. So two types of these passive transports, the first one being simple diffusion. That's where molecules are passing through the phospholipids directly, moving from high to low. These are nonpolar molecules and they are small in size, like oxygen. Facilitated diffusion is the second one that you learned. Facilitated meaning that it needs the assistance of a protein channel. So these molecules typically are larger in size and they are polar. Therefore, they have to pass through that protein channel that is embedded in the phospholipid membrane. All right, I'm going to move my little profile over here. And the next one that you just learned is active transport, which is actually moving molecules against the concentration gradient. So moving molecules from an area of low concentration to an area of high concentration. Because it's going against the concentration gradient, it does require energy of the cell. So molecules are passing through specialized protein pumps, sometimes known as carrier proteins, and these pumps are only allowing molecules in and out through that concentration from low to high, only if energy is available. Another type of active transport is bulk transports. So bulk transports also are using energy, but they are using vesicles to move many molecules at one time across the membrane. So there's two ways they can do this. The first way is endocytosis. This process is bringing molecules into the cell. And then exocytosis is bringing molecules out of the cell. So both of these are types of bulk transports, and again, they fit under the active transport umbrella. So the third type of passive transport that we're going to be learning about is osmosis. So osmosis is a special type of passive transport that is only focused on the movement of water molecules. So go ahead and take out page 10 of your packet, and we're going to fill out the first two boxes together in this screencast. So what is osmosis? Osmosis, again, is a type of passive transport. I'm going to move my profile thing so that it is out of the way. It's not moving. Okay, there we go. All right, so passive transport is the diffusion of water through a cell membrane. So again, we're moving from high to low concentration gradients. In this picture over here, you can see that we have our water molecules outside of the cell membrane. There's a lot of them. Therefore, we can infer that it is a high concentration of water. And then on the other side of the phospholipid membrane, we also have some water molecules, not as many, therefore we can assume that this is a low concentration of water. And with water molecules, it is very similar to the process of facilitated diffusion, where they do have to pass through a protein channel. This is a specialized protein channel that only allows water molecules to pass through it. Water molecules being polar, they cannot go past through that phospholipid membrane. So they do have to go through that channel. As a result, these water molecules are moving from high concentration to low concentration. So water is moving from an area where there is more water to where an area that there is less water. That's the high to low concept. And it is a type of transport passive transport that does not require energy. So keep that in mind. Let's take a look at another diagram so we can kind of see how osmosis works in action. So here we have a representation of a cell. As you can see, this pink dotted line, that is representing the selectively permeable membrane. So selectively permeable means that only certain substances are allowed to pass through. In this case, water molecules only. So we have on the left, we have some of these smaller blue dots. These are representing water molecules, and we don't see anything that's green on this side. If we look on the right side, we see that there are also water molecules and some green ones too. These green larger molecules represent a solute molecule, such as sugar, salt, or electrolytes. So if we look at this text on the bottom, let's focus on the water first. On the left, we have a high water concentration, and then on the right, we have a low water concentration. Remember, water wants to move from high to low, or where there is more water molecules to where there's less. So water is going to be moving to the right, as we can see by this arrow. Now, what about these green solute molecules? Notice that they do not pass that membrane. That's because the cell membrane is selectively only allowing certain molecules to pass through. So in this case, the sugar and the salt, they're going to stay on the right-hand side. You'll notice that because the water is moving towards the right to an area where there's less water, we can also see that this is a high concentration of sugar. Where the water originally came from, it's a low area of sugar concentration. So they're kind of the reverse of each other. So keep that in mind when you're looking at scenarios about osmosis. Water molecules are always moving from high to low, but they're going to where the most solute often is present. So in this case, the most solute is actually on the right, and that's the direction that the water molecules are going. All right, so that is the end of this screencast. Stay tuned for screencast number 2, where we're going to look at some examples and add them to our page 10 of our packet. Thanks for watching.